Hi, everyone, and thank you for coming today to talk about lumping and splitting, as well as looking and talking about Mondo. Let me change this real quickly over, and I will start to get my slides up. This is not just a gene activity, but it is very much related to that, but we welcome any of you that come from different curation activities to participate. Hopefully this will be a slightly fun session. I do hope that we can make it a little bit interactive and make it fun in that way. So for those of you that have your iPhones or have your computer out, as this is virtual, go ahead and pull those out for a QR code reader and or enter in this URL. We're going to have a little bit of polling questions that hopefully we're going to see live. It might turn out to be a disaster, but we'll make it through just in case. So with lumping and splitting, traditionally genetic diseases have been characterized based on the inheritance patterns and the phenotypes or collective phenotypic features of an individual or groups of individuals. But with the advent of technologies to quickly identify the genetic basis of disease, a predicament of multiple disease entities was given with any given gene. And as you can see by the examples that flashed on your screen, this is not limited to just one area or one clinical domain. It really is present within all. But specifically with gene disease relationships, we do want to focus on what the gene is telling us about disease, which poses a question: which disease entity do we curate when any given gene can be associated with multiple entities? And so again, that predicament becomes when to lump or when to split. Right? We know that genes can be associated with several phenotypes or phenotypic features, and how can we assess if the condition they are curating is the most appropriate? Why is this important, you may ask? Well, because the way that we define that disease entity helps us to figure out what types of evidence are included or excluded within a particular curation. And as you all know, because our frameworks are systematic and they are also on a point-based system, that could significantly change the classification that is reported for that particular gene disease relationship. And so if all curations are split for each phenotypic feature reported in the literature, we really could risk an infinite amount of curations on ClinGen that really may not reflect the true nature of each condition or association, nor that mutational spectrum of the gene. We would just have exceptional numbers of curations here. But on the flip side, if we tend to lump everything that is an existing phenotypic feature for a gene into one greater known syndrome, we do actually risk losing some intricacies of each condition and the phenotype or feature and potentially a clinical management. So it really is becoming into a balance of when to lump and when to split. And in order to address how we determine this, we developed a working group that was made up of experts in the areas of genetic disorders, nosology, ontology, as well as biocuration. So I want to thank ahead of time the whole lumping and splitting work group and the membership for the work that they do to help with our gene curations. So I do want to start off with lumping and splitting that we do have one general principle or overarching principle. And that really is that genes associated with a single published disease entity should only be curated for that condition, or what we would refer to as lumped, unless there are indications to split specific phenotypic features of a syndrome or variable phenotype into separate curations based on the guidance provided by criteria, and we'll go over that. So for instance, the gene NF2 with the very well-known disease neurofibromatosis type 2. We would suggest the NF2 is curated for neurofibromatosis type 2, rather than saying NF2 for Lish nodules. Lish nodules is a feature of the full syndrome of neurofibromatosis type 2. So this is the indication of that overarching principle when we think about syndromes, and then we think about features or constellations of those features within a particular disease. So as we said, we did develop some criteria for us to be able to determine what is the most appropriate disease entity, again, when we know those genes have several different phenotypes that are asserted in the literature or features. And so the first one that we really want to look at is the assertions, or what are the current definitions of that disease entity for that gene. So this is looking at what the literature has reported when we think about PubMed and those scientific articles, but also looking at other nosological and ontological authorities such as OMIM, even going into Orphanet and Mondo, looking at rare disease and different databases on NIH. but also looking about the gene's association with disease through those different authorities. The second one, and what really helps to drive our lumping and splitting criteria, because we are called ClinGen, the gen for genomics and for genetics, is the molecular mechanism. So we really want to appreciate when we think about these different phenotypes associated with disease, whether or not there are differences in the mechanism underlying those assertions. This could be things such as a loss of function variation within one phenotype or feature versus a gain of function on another. That would constitute a difference in molecular mechanism that may justify a splitting of two phenotypes to have two separate curations. We can also think of things like different transcripts as well as different domains. We know that there are several types of genes where you have a limited domain or even an exon with variations that results in a specific phenotype. Next, we wanted to appreciate the value of phenotypic variability and that expressivity among individuals. So really trying to look at whether or not the phenotype or the feature that is being observed segregates consistently within a pedigree when we think of individuals that have the same variation in that gene, or if they present similarly in two or more unrelated programs. So now that same genetic variant is now found in two different people, do they present similarly in their features? Or is there really variable expressivity, which we know can happen in the majority of individuals anyways, but really seeing whether or not those phenotypes between family members or unrelated individuals are really disparate or can have several different features with that same underlying variant. And lastly, we wanted to appreciate the inheritance pattern and whether or not there are differences in the inheritance pattern between those disease entities of interest. But we also need to consider in some aspects of whether or not they represent distinct entities, or do those inheritance patterns rather represent a continuum of disease. You can think of these as somewhat of dosage impacts as well, in which case we would think of semi-dominance. Some examples of that are when we think about LDLR and familial hypercholesterolemia. And so these are our 4 criteria, and as you can see, there's 4 of them. And so it really becomes a balance of whether or not you would take certain phenotypes and lump them together, and then they would create a new curation, or whether or not any of those features favor a split and they would be an independent curation. And so it's really, as an expert panel, you looking through these particular criteria and performing a pre-curation, which is where you accumulate evidence on each one of the phenotypes associated with your gene of interest and begin to bring back some high-level information on each one of these criteria. And so it really helps the expert panel, especially in situations where you may have 2 that favor a split and you may have 2 criteria that favor a lump. And we know that generates a lot of discussion. So there's certainly some opinion that goes into that. So hopefully we'll be able to get started. So again, pull out your QR code readers and or look at that website and see if you can start the poll here. And so I will just hopefully get this started. But I really want you to be able to tell us, has your group ever performed a pre-curation in your GSEP? I'm just curious, for the number of you out there, how many of you have actually done this within your particular groups? Great. And so certainly more of you can get on, and you can have other ways to participate with other poll questions in the future. So what's nice is we can tell that some of you really have already gone through this pre-curation process, which is great. You're really going to be informed about what it is, but certainly we have some of you who have not. So this high level overview hopefully will give you an introduction, and we can certainly be able to give you more within your different group. I'm going to move on now just to the next question. When we think about that, we have done lumping and splitting, and now you may have features that your group has decided were part of an actual disease entity. Have you in your GSEP had to define a new disease entity? In other words, have you had to create a new Mondo term for your gene curation so that you can move forward with that gene disease relationship and get a classification to publish it online? Great. Awesome, they're taking up there. So this is really great to see. Again, some of you are really familiar with this process. Some of you may not be yet, but we're going to introduce one of the issues that comes, as you can tell from this question, When we think about lumping and splitting, and we've now been into a situation where there may be 3 phenotypes that we lump, but there's a 4th one that is split. We are now dealt with having 2 separate curations. But when we lump those phenotypic features together, how do we really go about defining what that disease entity is? And beyond that, thinking about what you name the disease. And so we realized that with lumping and splitting, while it's great and we can think about these features that are being brought together and we're redefining or defining these diseases, that we've also brought on a predicament for our expert panels on how do you name this disease. So I just want to go over some of the typical options that occur right now when we think about every day that there are new gene disease assertions that are being made. Even still, we have 30,000 genes in our genome, and we have at last time when OMIM published about 15,000 of those associated with disease. So that leaves about 15,000 left that may or may not be associated. And again, whether at a monogenic or a polygenic risk, that's yet to be determined. But currently, the different naming schema that we go through for disease nomenclature is going to include eponymous names. So some of those very well-known ones that you might be familiar with are Marfan syndrome, Charcot-Marie tooth. It's where we essentially name this disease after somebody who either discovered that disease or somebody who published very highly about that particular disease or syndrome. What's nice is it does give credence and acknowledgement to the individual, but as you know, sometimes there could be acknowledgement that is also missed with having an eponymous name. The next one is a phenotype or a phenotypic varied name. So this could be things where you use Latin-based names. One of the oldest diseases we know that has the name is diabetes mellitus, which literally means a sweet tasting urine, or even retinitis pigmentosa, right? We're learning about the different types of phenotypes that are observed, or that most common one. You can also have clinically based names such as limb-girdle muscular dystrophy and amyotrophic lateral sclerosis. Some of these can tell you about that particular phenotype observing. Some could even be named after pathological findings that you have from different diseases if you're able to look at the pathology. Another name that you can think of, and that's used very highly in certain areas such as metabolism, is biochemical names. So these are things such as phenylketonuria, vitamin D deficiency, or even RASopathies. When we think about there's a name for a disease entity based on biochemical pathways or a biochemical function for a specific gene, and most likely for a family of genes. You can even think of things when they're families of enzymes together. You can also think about an acronym, right? So some famous ones or well-known ones are Me, Loss, and Charge. And while these are great, having an acronym, do you always remember what each of those acronyms in the phenotype stand for? Another predicament that can come with an acronym-based name is that if another phenotype becomes asserted or prevalent as you begin to discover more and more individuals with variation in this gene, do you begin to add an additional letter? And you can tell how that could be a very precarious incidence that happens depending on what's the next letter that gets added to the end of a word. And lastly, you can have a gene-based name or a gene phenotype name. One that you may think of right at this moment would be P10 hamartoma syndrome, where you really are trying to give a reflection that this is a genetic condition and that you want to explicitly say the gene, but also begin to combine a phenotype or a phenotypic feature that is most well known or most associated with that particular disease in those individuals. So we're going to go through a little example here. Now I will state, and it's down here, that we're going to look at CAV3 or caveolin-3. So if you're wanting to look along with us and go on OMIM and look at the different disease assertions, I will note that this is a picture taken from 2019 when this gene was first reviewed. It may have changed since then. OMIM changes all the time based on different assertions in the literature and or even what we bring back from our particular working group, and we work very closely with OMIM and the lumping and splitting working group, as well as ClinGen as a whole. So I just want to give that disclaimer here. But when we think about that pre-curation process with lumping and splitting and defining the disease entity, that first criteria really is to look at what are the assertions in the literature. And so we do direct our curators to go to OMIM and other resources such as OMIM, because that's pre-curated data in itself, right? Someone has already gone through looking through this data, and that helps to save time. So what we see is that there are 6 assertions at the time in 2019 that were given for caveolin 3. These may include a familial hypertrophic cardiomyopathy. It can have elevated creatine phosphokinase, a long QT syndrome, and some muscular dystrophies, and a mix of them, including a limb girdle, distal myopathy, as well as a rippling muscle disease that we have here. I can also bring you around to the criteria number 4 because we can take care of that here. And we can see inheritance pattern. And so again, through that pre-curation, we can see with the assertions, there are several different entities. They do display both cardiac and muscle phenotypes. And we do notice that most of them have an autosomal dominant inheritance pattern, indicating that it is predominantly monoallelic. But in the case of limb girdle muscular dystrophy, there may have been somebody who was asserted as autosomal recessive. Now, once you get into the full curation afterwards, you can really begin to determine, or even looking at that paper and individual to see where that, is that a confirmed autosomal recessive, or are they dominant and just predicted to be recessive because parents weren't tested? But off the hand, we can already see that inheritance pattern is going to be similar. Now, when we move to our next criteria, molecular mechanism, and we really get into the nitty-gritty of, are there differences in the mechanism between each of these 6 asserted entities? A high throughput way to do that is to really go look in ClinVar or any other variant database, right? I want to respect that the gene that you are looking at and pre-curating may have its own variant database, whether it is included in ClinVar, whether it has its own through LOVD, or even an additional external resource. Try to look out, go Google search, and I always look at my gene and I say variant and database just to see what's out there and what I may not be seeing just from ClinVar alone. Sometimes you can get some really great evidence. But in going into ClinVar, you can really begin to look at what are those pathogenic variants that have multiple assertions and maybe not conflicting. But again, you want to bring into some of those conflicting. They may have some pre-curated data that will be important. But when we look at those different variants that have been asserted in ClinVar and in some of these papers themselves, and we look at where they've been asserted for each one of the phenotypes on this Venn diagram, we can see that there's quite a significant amount of overlap for 5 of these specific phenotypic features, right? So what we do begin to see is that long QT syndrome and the variants here have no overlap with any of the other ones. And at this point, that suggests that long QT syndrome likely has a different molecular mechanism, and that this should be split off and have its own independent and separate curation. And we'll definitely go through and talk about that a little bit later, but that would be the suggestion, especially because one other thing you may want to note while you're doing pre-curation about some of these variants is to go through NOMAD. Do you see any of these variations are too high of a frequency to be considered pathogenic? You might want to star or highlight those. That might be an indication to split to dispute. But now when we come back here, we can see that many of these others, especially those muscular dystrophy phenotypes where we're seeing a significant amount of overlap, and also with that hypertrophic cardiomyopathy or heart, that there are similar variations over the same amino acid, that this really may constitute a spectrum of disease, and that it can present differently among different individuals, depending on variable expressivity, which we'd have to look at with phenotype next. So again, moving into that phenotype area, this is one of the examples when we do want to look at a pedigree to see whether or not there is any variability within this family or intrafamilial variability. And when we look at this specific variant that was in CAV3, and it happens to be this R27Q variant, and these individuals that are both genotype positive and phenotype positive, we can see that there are a range of different phenotypes that are observed in different family members. It can go from them being asymptomatic in a carrier, which can happen in some diseases, right? It could be that they were not phenotyped very well. Sometimes they're deceased, or they just did not present with a very severe phenotype. And then we can see some that have limb girdle muscular dystrophy, rippling muscle disease alone, or a combination of those. So again, representing a spectrum. Now, this is not in any way to disrespect the fact that rippling muscle disease has its own characteristics and findings at the clinic level, and so does limb girdle muscular dystrophy, right? These individuals are going to be diagnosed by how they are observed when they go into the clinic. But because we can look at the genetic level and look at these variants, it gives us a little bit more detail about how these different phenotypes can emerge within individuals, and really, again, trying to get down to narrowing in what is that disease entity for the curation. So let's move on to our next question here. When we think about, we've gone through this pre-curation and we've gone through to talk about these different entities that we have. Oh no, go. There we go. For CAF3, what we just went over and all of those 6 phenotypes. And if you want to consider whether you would rather lump or whether you would split the phenotypes associated with the muscle. So this is excluding long QT, but just on the information we've given, as you an audience, would you think that we need to lump limb-girdle muscular dystrophy, the distal myopathy, the rippling muscle disease, the elevated creatine kinase, and even the hypertrophic cardiomyopathy? But we also recognize that some of you may feel that they all need to be split and be their own separate entity. Great, maybe I'll convince you, or you're just being very nice to me as the presenter. But these are the questions and considerations you have to go through, and these are the parts of your expert panel where there really will be involved discussion as you think of lumping and splitting. So if we go with what we've suggested from your feedback and from what we're going to the presentation, we're going to assume that we're all happy and we're going to move forward with lumping this disease entity. It doesn't mean we can't ever split it in the future when we have more data, but this will be our decision for right now as we move through this whole presentation. So now that we've given our decision to lump, we need to look at the range of phenotypic features and those molecular mechanisms. Right, and we did find there potentially are 2 independent curations, and we would say at least 2 independent curations. We're going to move forward with one curation represents that lump disease entity, and it's going to include the hypertrophic cardiomyopathy, that increased creatine phosphokinase, limb-girdle muscular dystrophy, excuse me, and the distal myopathy and rippling muscle disease. Whereas that second curation will represent that split entity consisting of the long QT. For the purposes as we move forward, we're really going to be focusing on this lumped entity because, again, the exercise here we want to think about is how would you name this disease? What are different points to consider as you move through? This is going to be important for the second half of this workshop because as you create these new disease entities and names, you're going to have to define them and also create new Mondo IDs potentially for them. And so this is going to really help to show you not only considerations we're going to present out to you for ClinGen, but also learning about how you can go about with Mondo, creating these new Mondo IDs so you can move forward with your curations. So let's go back to those options that we had on how to name a disease entity. Well, we could go with the eponymous name. And at this point, if we did it, we would be into this a situation of Minetti, McNally, Herman, Carbone, Hayashi dystrophy, because we can't just limit it potentially to one person. Each one of these disease entities was discovered or asserted by a different individual. So at this point, it would be really combining them all. Otherwise, though, if that gets too long and too wordy, as you can see that it gets, if we leave anybody out, then how are those people going to feel? I just want you to kind of take into the considerations we have to think about when we're going to choose a name for a particular disease. It's not just based on alone of what we feel, but we have to think about other individuals, the patients themselves who are diagnosed with this disease, even so far as thinking about insurance companies when we're thinking about gene therapies, right, for different genes that are coming through and how those diseases are named and individuals and patients are picked. If we move on to our next option, we have the phenotypic description. And again, due to the numerous phenotypes that we are now lumping together, there's 5 of them, it may be difficult to produce an accurate nomenclature. Now, I don't claim to be a clinician or an expert in the muscular dystrophies, but this was just my shot at it, was that it would be a muscular dystrophy with hypercreatine phosphokinase, with or without limb-girdle muscular dystrophy, with or without Tateyama type distal myopathy, and with or without rippling muscle disease. Now, as an expert panel, they can think about whether or not one of these features actually represents many of them. Maybe rippling muscle disease represents where you can have distal myopathy with a molecular dystrophy or a muscular dystrophy that's limb-girdle type together. But these are those considerations that you need to make and the group needs to make as they move forward. And does it really convey the disease entity of what you're trying to accomplish and the inclusion of the data that you have for that classification? Now we move on to biochemical. Well, we could go through with caveolopathy. And in actuality, caveolopathy was one of the proposed names through the literature for these CAV3 associated diseases. So it wasn't only us that thought they should be lumped. There were other people who published in the literature about this. But this may also present an issue and a predicament. The current proposed naming scheme, but this nomenclature suggests that mutations in all caveolopathies may produce the same subset of phenotypes. And there are other caveolins. There's caveolin 1 and 2 in the very least, and there may be others that are going to be discovered in the future. And at this point, caveolin 1 and 2 are not associated with the same type of phenotypes. Actually, in fact, one of them, I believe, is not associated with a disease or a phenotype at all. And so those are some considerations you have to give when you give a broader biochemical name. Does every protein or family member within that biochemical pathway give a similar phenotype? Now we can move over to the acronym. Well, when we think that we have limb-girdle muscular dystrophy, rippling muscle disease, cardiomyopathy, hyper-CK, and distal myopathy, the best I could come up with was lurched. And as you all may think, lurched doesn't sound very pleasant. Individuals with muscular dystrophy may walk with a lurch, so this could be a situation where it seems a little bit of making fun of individuals. And so again, that consideration of that acronym. Obviously, too, if at any point there is additional data to suggest that one of these entities is not part of the lumped entity anymore, you would be removing one of those acronyms, which would now change its meaning and potentially its name. Lastly, we're going on to that gene phenotype approach, which may be considered or called a dyadic-based approach. But this is where you would begin to represent the gene that is responsible for this when especially you have a genetic disorder. Obviously, there are disorders that are not gene-based, and one of these other options may be completely and totally appropriate for it. I just want to reiterate, this is for a genetic disorder that we know has a monogenic basis. And so you can also harmonize that with the phenotype or the major phenotypic feature that happens to be consistent among the most number of individuals with variation in this gene. And so in this example, we have CAV3 related muscular dystrophy. This would begin to tell you that there is a gene associated with it, CAV3, but maybe it needs to say the gene CAV3 genetic related muscular dystrophy, because there could be individuals that don't understand CAV3 as a gene. That's one thing we would have to think about when we take off our hat as geneticists or experts in genes and put on a different hat of someone who doesn't know. But it does incorporate those specific muscle phenotypes as well. So pull out those computers and those phones again. I want to know from you, what would be your preferred disease nomenclature for the lumped CAV3 disease entity? Would you go with lurched? Would you go with CAV3 related muscular dystrophy, caveolopathy, an eponymous name, or a phenotype driven name alone? Great, thank you all. We're going to move on. And it doesn't mean that any one of these is better than the other, right? It depends on the situation of your gene disease relationship and within your expert panel and your clinical domain. It doesn't mean because you decide for this particular gene disease relationship that it always has to be a gene phenotype based name. You could find that a phenotype based name fits what your category is or that curation that you're doing for a different gene or set of genes. It's really just to give you an idea and start to help have you think about the complexity and the issues that come around when you have to define a disease entity, just what it is, redefine it, or to even create a new entity because maybe something isn't existing that fully captures what you're trying to convey with your curation. So I just want to talk a little bit about some future aims that we have within the lumping and splitting working group. So we've actually formed a subgroup that is called the Disease Naming Committee. As you've seen from this practice and this exercise, we know we created a predicament for our gene curation expert panels to start with, but this could even carry over to other VSEPs as they start to curate genes that have been lumped or split. But it is tasked with developing some guidance for expert panels when disease nomenclature issues arise due to lumping or splitting. This guidance will be reviewed by key representatives for clinical domain working groups and GSEPs for feedback, and it really is going to be something similar to what we have here, points to consider. When should you consider a certain naming schema? When should you maybe have pause for different naming schemes? It really is, again, to be an inclusive thing that is going to work over many of our clinical domains. We also are going to continue to collaborate with OMIM and Monarch Initiative with the nosological and ontological authorities, right, on how we can harmonize our efforts on disease nomenclature. Nicole is going to tell you next about what Mondo has been doing, because it is a resource that combines many of these different ontologies and nosological sites and the different names that they have. So it will be exciting for you to hear about what they're doing in the future. But this really is useful to help establish the consistency among our groups and to provide up-to-date information that may directly affect nosology or the ontology of diseases. And ultimately that may affect patient care. So all of us are really committed to try to help make that transparency of our different assertions. I also want to let you know that coming into this phase 3, we will be working on publishing those pre-curations that you work so hard to gather the data and information on the ClinGen webpage. This will basically act as a supplement to those gene curations that are already being published. It's another way that we add a layer of transparency to our gene disease relationships and the amount of data that we have collected to make these different assertions. And so let's, again, for that one last time, get out your iPhone or get out your computer. What level of knowledge or confidence do you have using Mondo? As we're going into this next talk, it would be good for us to know whether or not you've used this Mondo resource a lot. Are you a person who has gone through and used their GitHub that has put on, put in a ticket so that you can create a new disease entity or change the definition? Or are you really just getting to know this resource? You're a new person to a GSEP, you've heard about Mondo because we use it, or you don't know anything about it at all. Great. So for those of you that are the novice that are coming here, this is going to be a great time for you to hear about Mondo as this resource and how to use it. With the time that we have left moving through, at the end of the workshop, we will certainly take questions. I'll start looking through the chat and answer what I can and flag any of those for questions as we move through later on for Mondo. But I think it would be really good for us to move on to Mondo as you've got this in your brain about creating these disease entities and how you can do that. But I just want to share with you that you are not alone. This is something that ClinGen has already been doing. So it doesn't matter if you're a novice, you will become highly confident in your skills of using Mondo in the near future as you work with ClinGen. We have several of our gene curation expert panels that have already used Mondo and used this in a way to be able to develop new disease nomenclature. This is just a small sampling of the work that has been done across some of our groups, but they are clearly working through to be able to define and redefine disease entities so that you can move forward with those curations and make it very transparent about what is being curated. I do want to take the time to thank you. And if you have not already gone through any of the lumping and splitting guidance, you can find that on the clinicalgenome.org website under working groups, lumping and splitting. And if you scroll down, you can get those curation guidelines. You can even look at a pre-curation template and an example that we have there. And I want to thank you all very much for your time, and I will kick this over to Nicole.
Thanks very much, Courtney. That was a great talk. So next I'll be giving an overview of Mondo. That poll is very helpful to see how familiar people already are with Mondo, and it seems like most people fall into the novice category, but several of you said you're comfortable with using it, so that's great. So hopefully there'll be some new information in here that can help even further familiarize yourselves with Mondo, or and for those who are newer to it, I hope this helps clarify some of the information about Mondo that you may not be familiar with. And yeah, so I am Nicole Vasilevsky, and I recently, as of Monday, just remotely moved to the University of Colorado, the Anschutz Medical Campus. I'm based in Portland, Oregon, on the West Coast, where it's going to be unusually hot this weekend. And so these slides are available at this bit.ly link here, the bit.ly ClinGen workstream. And yeah, I'm part of the Monarch Initiative and part of the Translational Integrative Sciences Lab that is led by Melissa Handel, formerly at Oregon Health and Science and Oregon State University. And now our entire group has remotely moved to the University of Colorado. So just an overview of what I'm going to talk to you about in these slides. So first I'll give you just a general overview of Mondo, some background, a little bit of background, kind of an introduction, even to some introduction to ontologies and why did we create Mondo, and what is it? And then some updates of recent changes that we've made to Mondo. And Mondo is a constantly evolving source since its creation, so it's always been updated. I'll give you some highlights of some recent updates that we've made, and then some information about how to request changes to Mondo. So Mondo is a community resource, and it's intended for use by the community and also contributions from the community. It's not just meant to be developed by us in our group. We want input and contributions from everybody. So I'll take you through the workflow of how to request changes to Mondo via our GitHub issue tracker. And then I'll talk a little bit about using gene-based names, which Courtney very nicely discussed in the last presentation, and how we want to address all the issues that ClinGen needs and the types of naming schema they want to use, that you all want to use. So I'll just give you a little bit of background on how to request these types of either renaming or names for Mondo terms, and then a bit about our obsolescence workflow. So terms are never deleted from Mondo. We try to keep, if a term is ever added to Mondo, we don't never delete it or entirely replace it. We use the kind of obsolescence process where we obsolete it and then replace it with new terms or point it to other terms to consider. And we try to keep everybody informed when we're doing this so it doesn't disrupt your systems that you're using. And one other thing to point out at the bottom of the screen is the link to our Mondo website. So you can find out more information about Mondo at this site here. And like I said, the slides are available at this link, and I think I have this link on every slide, or most slides. So you can follow along and follow along with the presentation. So just some background about how our group and what we do. So I'm part of the Monarch Initiative, which is an NIH-funded project that is it's building a resource that connects information about human phenotypes and genes and diseases with model organism information. So it's a queryable database where you can query for information about diseases and genes that are involved in connections to different model organisms. So like mouse models, zebrafish, etc. And so the Monarch database is available at the site monarchinitiative.org, and it uses underlying ontologies that allow for inferencing and query across the data. So Monarch has been around now for probably about 8 years, and the Mondo ontology was created as part of this effort. And just to introduce you to our team, so we're a growing team. Mondo, as I'll show you, was initially created by Chris Mungle, who is one of our semantic engineers, and I like to call him one of the gurus in ontology development in our ontology world. And he originally created Mondo, and he leads our development and ontology engineering team. I lead the curation efforts for Mondo, and we just recently hired a new curator, Sabrina Toro, who is on this call, and she will be helping with addressing a lot of the issues in Mondo. So she hasn't started working on any ClinGen related tickets just yet, but she's only in her 3rd week or so, but she's very quickly learning and is a great addition to our team. And so she may be someone that if you have specific requests to Mondo, probably me and her will be the people that you want to contact with any specific questions. And I also want to point out, if you make, I'll show you how to go through the process of asking for changes or requesting new terms. But I just want to point out that at the moment, we're a little bit, we've been a little bit under-resourced on Mondo, but now that we have Sabrina on board, and also we have Kalia, who's part of the European bioinformatics team. She's also just recently came on board to help with Mondo curation. So we're a little bit behind on our tickets at the moment, but now we have these two fabulous new curators that we hopefully will be catching up with some of our open requests. And then I'll say this and I'll say it again later, that always feel free to contact us and nudge us if you have any requests to Mondo that for changes or new terms and you want to know the status. We're more than happy to be nudged or nagged about it. Feel free to contact me or Sabrina and to say, hey, what's the status of this? And we'll move your tickets to the top. So our other really great members of our team are Nico Matanzoba, who does a lot of our quality control and development and engineering efforts in Mondo. He's been doing a lot of work to automate a lot of the processes in Mondo, which has been really helpful just in helping clean up a lot of Mondo. And Daji Zhao recently came on too on the engineering side. He's working with Nico very closely to help with a lot of the automated processes to make Mondo just a more consistent and run smoother. And then Peter and Ada are, a lot of you probably know Ada from OMIM. They are properly funded on Mondo and part of our team, but really, and they provide a lot of medical expertise for the types of terminology that we are adding, but the medical experts are not just them, they're all of you. So we are really grateful for all the input that you provide. And we really, our team is composed of, we come from very different backgrounds. Some of us are biologists, some of us are computer scientists. So we really need the contributions from all of you with your medical perspective to ensure that we build the most logically coherent resource. So yeah, we have a really great team and it's a really fun project to work on. And we're all, we all actually, I think all of us love it very much. So if you're not familiar with ontologies, I just like to always do like kind of a what is an ontology kind of background slide, because it's a very, sometimes it can be kind of a hard concept to describe or to understand. So I think most of you probably know that Mondo is an ontology and it's used for structuring data, but why use an ontology versus just use like a controlled vocabulary or like a thesaurus, like mesh or something like that. So an ontology is formally defined as a knowledge classification of a domain. So it's a classification of information in a specific area of knowledge. So in the case of Mondo, it's classifying information about diseases. But some of the key features of ontology is that the terms are defined both textually. So we have a term, like a textual definition of a term like epilepsy is a disease where patients have seizures. But then they're also, those terms are also semantically defined. So the relationships between the concepts are formally and logically defined. And this is one of the key parts of an ontology, is that you can do this logical inferencing and querying across the data, and you can do computational reasoning. And that's what really the power of an ontology is, what makes it justified why we would build an ontology instead of just creating like a list of terms that you can use to index data. So in ontologies, like any kind, like a lot of controlled vocabularies, the terms are arranged hierarchically, and then they are expressed in a computational knowledge language, such as the web ontology, which is abbreviated OWL or OBO or RDFS. So this is an example of just a hypothetical ontology that I borrowed from someone at a conference. This is a swantology. It's an ontology about swans. So you can see in this bright red color, this swans are a type of bird, and then you have subtypes of swans, like you can have tundra swans and black swans. So this comes from the NCBI taxonomy. And so these terms are kind of hierarchically arranged. So every tundra swan is a type of swan. But also the power of ontologies is that you can have these relationships to other classes or other terms. So this blue hierarchy here is coming from Uberon, which is the Uber anatomy ontology. The name was originally conceived of as a joke, and then it stuck. So now we have Uberon. We also have Uber Pheno, which is the Uber phenotype ontology that aggregates various phenotype ontologies. And in Uberon, you can see that, a swan can have a part that's a web foot, that's part of that is a foot, that's a web foot is a type of foot, and a foot is a type of anatomical structure. So we have this like hierarchy from Uberon, but we can use these relationships between the swan's web foot relate the swan's web foot to the organism, the taxon description swan. And we can say the swan has different capabilities. It's capable of these biological processes like swimming behavior, which is a biological process that comes from the gene ontology. So ontologies are really powerful because you have this big kind of knowledge graph and knowledge representation where all of these relationships are formally defined using this computational logic. And then you can do a lot of interesting querying and analyses over the data. And this link here at the bottom points to a web page from the Obo Foundry, which is a organization that defines and provides guidelines for ontology development. And several ontologies are part of this foundry where we commit to building ontologies in an open and accessible format and adhere to a shared set of principles. And on this website, on the Obo Foundry website, we created a web page that has a bunch of resources about ontology. So if you wanted to learn more, there's like some links to articles and tutorials and to different software applications that you can use. So if you wanted to get more into detail about ontologies, I recommend looking at this page. So, so when it comes to disease ontologies, we created Mondo a number of years ago, kind of with a question of what would be the most clinically useful way to define and group diseases. So prior to Mondo, there was a lot of different terminologies that existed and still do exist that cover different kind of subject areas or sub areas of disease. Like there's ontologies that are terminologies that are dedicated to cancer, like the National Cancer Institute thesaurus, or complex diseases or rare diseases, like Orphanet covers rare diseases. There's terminologies that specialize and focus on infectious diseases, which are probably particularly useful now, thanks to this pandemic, and Mendelian diseases like OMIM. So we needed we needed an ontology that would span multiple categories that covered all these different areas of disease in order to properly be able to aggregate the data in the Monarch initiative. And we needed a systematic way to relate these concepts. So a lot of these terminologies and ontologies that currently exist or existed at the time have mappings between the terminology. So say a term from NCIT may map to another term from Orphanet, but there was a problems with these mappings where they weren't always consistent. So a mapping between one terminology and another one didn't always mean that the terms were necessarily equivalent to each other, and the semantics or the actual meaning of the mapping wasn't always defined. And then once you have mappings from one terminology to another, and then that terminology maps to another terminology, then you end up with n to the squared sets of mappings. It just can grow exponentially. And there's a problem too of where there wasn't always a one-to-one equivalence. So sometimes a mapping would map, a term would map to another term in another ontology, but then it may have just been a related term or maybe it's a subclass relationship, but it wasn't clearly defined. So it wasn't impossible to know exactly if we tried to aggregate all these different, or tried to use all these different terminologies and ontologies separately, we didn't actually know precisely how they were relating to each other. And Melissa and several other folks from our group did a review paper a number of years ago where they did a census of disease ontologies. And in this paper, I believe they found something like 30 or so disease ontologies and terminologies that currently exist. And we can see kind of the problem here where there's all these different terminologies that exist, but which one do we use? Which one's the best to use? Actually, in another project, I did an extension of this, just kind of looked at if I could compile a list of all the disease ontologies that I could find. And I came up with a list of, I think, 46 different terminologies, including Mondo. But so yeah, so there's been this proliferation of disease ontologies, and which one was really the best to use was a question for us before we started Mondo. So yeah, so there was this wealth of resources, and some were very specialized, some were more generalized, but no single resource seemed to be sufficient to fit our needs. The resources that were too specialized didn't always include all the concepts we needed. So using something like OMIM is a really great, well-structured, well-researched resource, but then they specialize just on Mendelian diseases. And then something like MESH is more of a, MESH is not actually an ontology. So how do we ontologize that? So yeah, and some of the generalized ontologies just lacked the precision and the depth and the key domains that we needed. So that's what led to the creation of Mondo. So Mondo is an aggregated resource that aggregates different source ontologies into this one coherent, precisely mapped ontological resource. So on the left-hand side here, you can see that some of the source ontologies that we integrate into Mondo, I think in total we have less than 20, about 18 or so sources that we use to seed Mondo. So we use resources like Orphanet for rare diseases, the National Cancer Institute thesaurus we use the neoplasm branch. GARD is an NIH resource of rare diseases, the experimental factor ontology, the disease ontology. We map to the ICD-10, which you may know is mainly used for indexing and for coding medical encounters. We map to MESH and we use OMIM. So we aggregated all these source ontologies, and Chris wrote this algorithm called KBOOM that does some kind of logical and probabilistic inferencing. So it takes two classes from the sources and compares them to each other, and based on the syntax of the label and also based on the logical definitions, it assigns a probability score, what the probability is that these two terms are equivalent to each other, or these two terms are superclass or subclass relationships, or parent and child. So that was how Mondo was initially created. And then we did some curated, some iterative curation over the initial product of the algorithmic combination integration of the terminologies, which is what I was doing. And then initially it was intended that Chris would run this algorithm, I would manually review the results and kind of look at the probabilities and say, based on this is a high probability, so this is probably okay. But then I'd look at the low probability and say, okay, these terms seem like they should or shouldn't be equivalent to each other. And then we would go through and run the algorithm again. We iterated over Mondo in that way for initially for the first year or so, and then now we just have Mondo as the resource, and it's purely manually curated now. So in addition to all the source ontologies too, we, as Courtney alluded to, we are adding new terms to Mondo routinely too, a lot of them coming from ClinGen suggested new term requests. So like I said, Mondo, the terms are all the terms in Mondo are given Mondo IDs, and then there are cross references to the other underlying resources where we obtain these terms. So we use these logical axioms, explicit logical axioms, to define the relationship between the terms. So if we have a term like mucopolysaccharidosis type 1, we would say this is an exact match, or in the OWL file, let's say equivalent to, then it would be equivalent to like NCIT, equivalent to Orphanet, but then it's to the DO term, or it's a close match to the SNOMED term, so, or it's related to. So we are able to define if a term is exactly equivalent to the source term, or if it's just merely related or a subclass of relationship. So this is unique to Mondo in that we have these mappings that are precisely mapped and defined. So now all of our users can know if a Mondo term is exactly equivalent to one of these source ontologies or terminologies. So, so the current status of Mondo is that we have about almost getting close to 25,000 disease classes, and each of these classes, like I said, is assigned a Mondo ID. But then we have the cross references to all the underlying ontologies. So almost every term in Mondo is cross-referenced to another terminology, unless it was just created de novo for Mondo. We use standard ontology engineering practices so it's interoperable with other obo-foundry ontologies like the human phenotype ontology and a lot of other obo ontologies. We do periodic alignment and sync with our existing resources. We go to some of the sources and try to update the sources. Like we monthly we'll go to OMIM and look for new terms that are added to OMIM and add all the new terms as OMIM as new terms. We'll go in and add those to Mondo, and then try to sync with the existing resources to make sure as the obsolete terms, we can keep those up to date as well. And then Mondo is released on a monthly basis. So around the 1st of every month, I do a release, and it's available in OBO, OWL, and JSON formats. And to date, we've had 61 releases, and we're getting towards the end of the month. So I'll be doing the next release in about a week and a half from now. Another thing I should point out too is that the monthly release schedule is just kind of my decision, or our decision, that we just release monthly. If any ClinGen user such as yourselves needs a term to be available sooner than on a monthly basis, you can absolutely ask me and I can do a mid-cycle release, and that's no problem. It's actually pretty straightforward and quick process to do the release. So if you needed something sooner than the next month, then feel free to reach out to me and we can likely make that happen. So yeah, so we have over 20,000 classes, and with our unique classes that are not obsoleted, we have about 22,000 classes. About 70% of them are defined. A lot of the definitions come from the source ontologies. So if classes are undefined, it's usually because they were not defined in the source ontology. Every time we add new terms, I can't think of a single case where we ever added a new term without a definition. So yeah, so the undefined terms are really just because the source ontologies haven't defined their terms. In some cases, we've gone ahead and added definitions to terms, but this has been something that we definitely would like to do, but it's less of a priority for us. So maybe with our increase in resources as of late, that's something we could increase the number of terms with definitions. And yeah. So we define this, and I'll go into this in more detail later, but we define the synonyms based on whether they're exactly the same meaning, which is an exact synonym, or related, broad, or narrow. So the majority of terms have some kind of synonyms annotated to them. So where can you view Mondo? So Courtney showed some screenshots from the ontology lookup service. So there's several places that you can look to see the terms that are in Mondo. I like to go to the ontology lookup service as my go-to when I'm viewing Mondo. It has a very nice layout. So this is a screenshot of an OLS page. The website is available here at the bottom of the screen, or if you just Google ontology lookup service, you'll be able to easily find it. So the OLS shows the term definition, the short ID, which is called a query, or the long IRI, which is here. You have the summary or the text definition here, and you can see the link to the source. This is where this definition came from. So this is the definition from the NCIT that we're reusing. You can see the hierarchy displayed below the term information. And you can see that Mondo is a polyhierarchy where we have terms as children of multiple parents. So we don't just have like a single classification. We have this multi-classification. So this term mucopolysaccharidosis is a child of rare disease with glaucoma as a major feature. It's also a child of congenital nervous system disorder and developmental anomaly of metabolic origin. And you can see some of the term information in this term info box. So you have all the cross references displayed here, and you can see some of the equivalences here. And then also as well down here, you can see the close matches, which reference SNOMED terms. And then we use some subset tags in Mondo, and I'm going to give you an example and later on in the slide deck where we now have a ClinGen preferred subset tag. So this we still need to work on applying this to all the ClinGen preferred terms, but ultimately, once this project is done, we'll be able to get a subset of Mondo that's the ClinGen preferred terminology. I'll talk about that more towards the end of the slide deck. And then other places where you can view Mondo, also going to the OVO Foundry page that I mentioned, the OVO Foundry earlier. So this page has links to all of the release files and other information about Mondo. You can also go onto our GitHub website and actually download the our OVO files directly from GitHub. And you can view it in this ontology software Protege. If you're familiar with using Protege, this is kind of my preferred way to look at Mondo, but it's a little bit unintuitive if it's not something you're familiar with. So the ontology lookup service is probably your best bet. And yeah, so Mondo is a community resource, and we really love having input and contributions from groups such as yourselves. And so yes, we use GitHub as our place where we store the ontology files, but we also have our issue tracker there. And we've had over 1,500 issues reported to Mondo since we started this project. And yes, we try to keep on top of them, but there are quite a few issues on the tracker right now, and we're working really hard to address these. And we hope to make some headway on getting some of these tickets closed. And I know there's some open tickets from ClinGen right now, so we'll definitely be prioritizing those and hopefully getting those closed out before the end of the summer. We also have a users mailing list if you wanted to get updates about when the releases are, whenever I do release, I send out an update to let everyone know that the new release files are out. And then I send out information about when we're planning to obsolete terms from Mondo as a warning and as a opportunity for people to provide feedback. If there's any issues with certain terms being obsoleted, we want to get feedback and any kind of concerns before we proceed with that. And I'll talk a little bit more about the obsolescence workflow at the end. And then, yeah, you can contact me by email anytime or via my GitHub handle on GitHub. And yes, we have weekly calls that are on Thursday mornings. So mornings are my time on the West Coast, or afternoons on the East Coast for most of you. And everybody's welcome to join those. We usually go through open tickets and talk about open issues in Mondo. So if that's something that you're interested in, you're more than welcome to join. And also, if you have specific questions, like if you have a ticket that's open and you want to discuss it with us, we can add that to our agenda and prioritize that on any week that you may be interested in joining. And we're also trying to hold more workshops with Mondo. We have a workshop coming up in July where we're going to be talking about some data modeling for common diseases for the Monarch Initiative. So that's something you're interested in, feel free to contact me, and we're more than welcome to join. So some updates of what we've been doing on Mondo lately. So yes, we do have all of our tickets that keep us very busy, and that is one of our biggest priorities is addressing the user needs. But there's a lot of known issues with Mondo too. Like I said, Mondo was created by aggregating all these resources together using this algorithmic approach, which is a very sophisticated and a really cool way of seeding the ontology, but of course it wasn't perfect. So there's a lot of known issues in Mondo where we're trying to go through and just refine and revise and improve the classification of all the branches in Mondo. So one effort that we've worked on recently, and that Sabrina, who's newly joined our team, is focusing on, we had a workshop in April that some of you were able to attend, where the focus was just getting our team together and talking about the top level hierarchy in Mondo. So the highest level class in Mondo is disease or disorder. And this is our previous classification of Mondo prior to the workshop. And this is kind of the classification that came out of the initial development of Mondo. But there had been some criticism that this was not a very intuitive hierarchy for browsing the hierarchy, especially coming from a clinical perspective. So per that feedback, we had a workshop and spent several hours virtually talking about how could we improve this hierarchical classification, and how would we want to see this top level hierarchy be displayed in Mondo. So one of the approaches that was suggested that we do was use just a medical textbook and think about how does a medical textbook organize the different diseases that that occur in human biology or even across different species. And we actually looked at, I don't know how many of you are familiar with the Harrison's internal medicine textbook, and we took that textbook and we looked through the different chapters and how they classified information about diseases, and tried to create a new proposed hierarchy based on that. So previously there was a lot of overlap between the different types of top level information. And this is because, like I said, Mondo is a poly hierarchy where a class can be classified in more than one way. It can have more than one parent, but we hope that this revised classification will be more intuitive for clinicians and for all of our users. So this is something that we're currently working on updating, and it's a bit of a big project, but it's something that is currently underway, and Sabrina's taking the lead on this. So as we work on this, we'll definitely be, there's already a lot of new tickets on the issue tracker. If you're interested in any of these particular branches and would like to provide any feedback, we'd be very grateful for that. And yeah, we'll be communicating this with the users via our GitHub repository as we go through and make these revisions. So this is something that we hope to get done over the next, this is one of our big priorities to get done over the next several months, we hope. So here's just an example of a more specific branch that we paid particular attention to, and this is based on an issue that was reported on the Mondo issue tracker. So we had this classification of cerebellar ataxia, but it was pointed out that not all ataxias are cerebellar necessarily, cerebellar ataxia being a movement disorder. And so we went through and looking at the medical text and looking at the literature, we went through and revised this classification. So we created this new class called atactic disorder, which is a child of a central nervous system disease. And then we classified these by either acquired or hereditary, and then also by mechanism, where we have either cerebellar, sensory, or vestibular. So some of these classes hadn't already existed, and the classification didn't seem to be entirely accurate. So this is an example of an effort we did recently to try to address some of these incorrect classifications. So yeah, there's a lot of known issues of Mondo, just and subtleties like this. So we're definitely making a lot of effort to try to improve these, and we definitely really value any user feedback in any of the hierarchies or branches in Mondo, and we'll work to the best of our ability to properly classify this information. So that being said, I'd like to just go through some of the steps for requesting either new terms or any changes to Mondo. So if you're working as part of one of the GSEPs and need to request a new term to get a new Mondo ID for your curations, or if you just see any issues in Mondo, this is the workflow that we use for users to request any changes to Mondo. So on the left here, I've outlined the steps that are needed to request a new term, and then I have a video which I'll play on the right that just shows, this is a screenshot of the Mondo GitHub repository, and I'll show you the steps that are needed to go through and request new terms. And so first I should point out that you will need a GitHub account in order to make any requests on the Mondo tracker, but GitHub is free to use. So if you just go to github.com, you can sign up for a free account, and then you can, everybody can access the Mondo GitHub repository. It's free and open to everybody. So, so if you go to this page on the Mondo repository, and then you click on the issues, if you click here, you would go to this page and click on the issues, and then you click the new issue button. And then we have a number of templates that specify the types of terms to be added, and then you can fill in the information that's requested. So we have we have a template for adding new diseases that are specific to diseases. And then there's some headers and already added to this box here where you can add the preferred name and ask for synonyms. So you can specify synonyms that are needed for that term. We also ask that you include the parent term, the ideal parent term for this term. And you don't always have to know all the answers to all of these things. If it's unknown, feel free to leave it blank. The only benefit of adding all this detail is it just makes it easier for us, the curation team, and we'll get to your request faster if all this information is included, particularly if it's in this format. But it's definitely fine if not all this information is given or if it's unknown. We are happy to look at this information if needed. So with all the definitions, we also include a source, usually like a PubMed.
Sorry, I just want to break in quickly. Can everyone please mute? Make sure you mute. I'm hearing a lot of keyboard noise. Thank you.
Oh, it's a video actually.
It's in the video. Got
Yeah.
it. Okay.
Yeah.
Sorry about that. Okay.
No problem. Yeah, I know. I didn't turn off the sounds on the video when I was recording it. And then also we ask that you include an ORCID ID if you have one, and then we can include that in the record. And then also it's really great if you include your the link to your ClinGen working group, and then we include that on the Mondo record too, so you get credit for adding a new term. And then with using these templates, it's really nice that I will get automatically tagged, and then it'll automatically be labeled as a new term request too, if that's the template that you use. And then I'll also be emailed and notified that there's a new term request there. So I'm getting all kinds of notifications, and I'm aware of all the tickets that are being issued. And then, like I said, we're a little bit behind on our tickets at the moment. So if there's a ticket that you added and you are waiting for your new term to be added, feel free to tag me on the ticket or send me an email and nudge me, and I'll be happy to prioritize that. So these are some recommendations for and best practices for writing tickets and new term requests, but these are just recommendations, and I just want to emphasize that we really appreciate all of your tickets and your issues that you report and your contributions. It's immensely helpful for extending and improving Mondo. So these are just some recommendations, and by no means do they absolutely have to be followed. And if any of this stuff is missing or isn't properly formatted, it's absolutely okay. Just know that we're very grateful for your requests. But if just in case you want to follow some of the best practices, here are some recommendations here. One thing that we request is that you should check to make sure the term that you're looking for doesn't already exist in Mondo. And just by checking like an OLS is usually a good place to look. And I will check that too, but that's one thing to do. And then maybe if it already exists, maybe it exists as a synonym, then you don't need to request the term. If possible, providing a concise definition in the definition field is very helpful. And I linked to an article that was written by Chris about some guidelines for writing good text definitions. If you add synonyms, maybe it's very helpful to provide a cross-reference to the synonym. In Mondo, we cross-reference almost all of the annotations that we add on the terms. And if a cross-reference isn't provided, but it is provided for the definition, I usually just assume that definition cross-ref applies to the synonyms as well. And then when requesting new gene-related terms, in particular, if you could check OMIM to see if there's any existing diseases that have that gene implicated, that maybe the term you're requesting is a grouping term that can group together existing OMIM classes. And then these formatting things are really just very minor but helpful if you're mindful and are able to adhere to some of these formatting guidelines. So all the terms that go into Mondo are lowercase. So unless it's a proper name or abbreviation, so writing the labels and synonyms in lowercase is helpful. The templates are written in Markdown format, so that's why they have the weird asterisks around all the terms. This is when you hit submit, it'll show a bold the top level field. So just write your request below that. This is very minor, but very helpful. Just to note that the PubMed IDs, when we add them to the ontology, they have to be in this format where there's no space. So when you copy and paste from PubMed, the PubMed has a space in the in between the PubMed ID colon and the number. So if you just delete that space, then it'll be properly formatted. When listing the parents, potential parents or potential children, adding the IDs and labels is helpful, and then putting them in a bulleted list is also very helpful. Just makes the process a little faster for me, just to enable me to copy and paste into the ontology. This is a paper on writing guidelines for writing definitions of ontologies. This might be way more detailed than most of you may want to know, but just in case you're interested in learning kind of how ontology definitions are ideally written, this is a resource for you. And then, yeah, so like we've talked about in ClinGen, the preferred naming is to use these gene-based names, and we're absolutely able to accommodate these. Yes, we have some templates that we use to write the gene-based names. So usually with the gene and then the name of the disease. And then a lot of times these are used as grouping terms for OMIM terms. And then we also write these logical definitions. This is kind of a computational part of the ontology, where we'll note that the disease has basis in dysfunction in some gene. So basically it says this disease is associated with this a variation in these particular genes. And then we have these different synonym types that we can add to Mondo. So they can either be exactly the same term, like familial Wilms tumor is a synonym for hereditary Wilms tumor. We can have terms that are more specific, more which are narrow, or more general, which are called broad, or related, where the term is used synonymously with the primary term in the literature or elsewhere, but may not be strictly correct. And then we also are able to exclude or deprecate terms. And this would be done in cases where we don't want to have that synonym. Like, for example, the NCIT has a lot of like cancer terms where it'd be like breast carcinoma, not otherwise specified. We don't want to have such general classes in Mondo, so we exclude these synonyms, or deprecated, where synonyms are no longer socially acceptable to be used. So we like to keep a history of all the synonyms that come from our sources, but then we don't always want to display them, so we can mark these deprecated. And then, yes, so this is what I wanted to tell you about. Another thing that's new is that we have these, we're now able to add ClinGen preferred synonym types to the synonyms. So this is something we just started doing, but we're going to work on an effort to go through Mondo and find all the terms. We're still determining the approach, but I'm thinking that we will go through all the terms that have reference to ClinGen through this clinicalgenome.org link, or I know most of you have submitted term requests to Mondo. I can look for your specific tickets and look for the new term for your terms that way, or look for the gene-based names. So ultimately, we'd like to be able to tag all the terms that are in Mondo that are the ClinGen preferred naming. And Nico, who I mentioned earlier, who's our ontology engineer, we're going to try to come up with a spreadsheet-based template where instead of having you go through GitHub to request synonyms or new term names, or you could just do it via like a spreadsheet template, and then that will automatically apply the changes to Mondo. All you'll see is a spreadsheet, and then he'll write some scripts that'll automatically apply the changes to Mondo, and then they'll be available in the next release. So we're working on some workflows to help improve our workflows with your team, to try to make sure that you have the term names that you need, and that that they're marked as ClinGen preferred, because I know there's some disagreements on how term names should be named. So in the cases of like this case, synostosis, craniosynostosis 4, this is an OMIM term. So I think our plan in Mondo is to, as a primary label, to use the OMIM labels, but then we'll be able to add exact synonyms that are ClinGen preferred for your use cases. And then once we've completed the application of these synonym type tags to all of the terms, and there should be a way for ClinGen to have like a ClinGen slim version of Mondo or a ClinGen version of Mondo where you can see just the labels that you want to see. So we'll be working with you on that. We'll be in touch with you to talk about how we can best implement this. And then I also created a spreadsheet in the case if you had a lot of new terms that you wanted to add, you could just go to this link and use this template and just add just in a spreadsheet format the new terms and text definitions and such, just basically the same kind of information that you saw on the GitHub issue tracker, but this is just a spreadsheet format. If you had a case where you had a lot of new terms to add, you're welcome to use this template, and then I can add the terms via this. And then finally, a couple more slides, getting towards the end. One of the last things to talk about is the obsolescence workflow. So like I said, the terms in Mondo are never deleted. We always retain all the provenance of all the terms that have ever been added to Mondo. But we want to make sure before we obsolete anything that we inform everybody and give everyone a chance to provide feedback. So on GitHub, we have labels that are marked either obsolete or merge. So either just entirely obsolete a term or just take it out of usage, or we obsolete and then merge it with an existing term. So if we have like duplicate terms, we'll get rid of one and merge it with another. So the workflow is meant to be that we mark all these tags on GitHub, or label all these issues on GitHub, and then email the Mondo users list. And if you're not on this list and want to be, please let me know. And then wait for at least 2 weeks before we take any action on merging or obsoleting these terms in the ontology. And then always in the obsoleted term, we'll add a replace by or consider. So the replace by is the term that's replaced the obsoleted term, the term that should be used instead of the obsoleted term, or consider if it's not a merge, say we obsolete mucopolysaccharidosis, and we say consider using another term instead. And then we also provide a reason in the obsoleted term. And this is a workflow that we just started relatively recently. So not every obsoleted term will have this, but every obsoleted term going forward will. So we have a reason, like the terms are merged or they're duplicates. And then we also are now linking the GitHub ticket to the term that was obsoleted. So you can go and see, go back to GitHub and see the conversation that occurred talking about why the terms were merged. And then the obsoleted terms will be available in the next release. So you have to say that there actually did see an incidence where when we bring in new OMIM terms, in cases where we have an OMIM term that turns into like a phenotypic series, so a new term, an OMIM term becomes a new grouping class, we do obsolete the original term and then create the grouping class and then the like type 1 and type 2 disease terms. And so that we were doing this workflow without doing this emailing the Mondo users list. And I noticed that did create some problems with ClinGen, where there was a term showing up in your database that had been obsoleted, where we basically just replaced the ID for the term with the label stay the same. So we'll make sure going forward that any term that's going to be obsoleted will notify the Mondo users list. So you have the opportunity to update your database before the terms have changed, to make sure that you know the terms have changed. And also when we send out the release, the Mondo release, there is a list of all the changes in Mondo with each in the release notes, so you can see the new terms that were added and any terms that were obsoleted. So that's another way to see all of the obsoleted terms each month. And then lastly, just this workflow, I just want to show you kind of like the timeline of what happens when you request a change to Mondo. So all the changes are reported through our GitHub website. And then there's a waiting period for community and expert advice from like our medical experts or from you. So everybody's welcome to give any comments or add to the discussion on GitHub. And then the curation team adds new terms to Mondo, usually with our cat sitting by our side, and creates a pull request. And then the pull request is then reviewed by our team and then merged, and then it's added to the Mondo edit file. And then the Mondo edit file is what's released around the 1st of every month. And then you can see, you should be able to see the terms in the ontology lookup service about a week after release. The OLS is updated every few days. And that is all I have for you. So thank you all so much for listening. And I think we have a few more minutes still at the end.